Hello, everyone. My name is Eric Thomas, and I'm a student in Biology 222, Human Genetics, fall semester of 2022. This video is a part of my semester-long project. The topic that I chose was to complete homework 1 in its entirety and do so in a way that's easily understandable and digestible, and then also create a YouTube video that was complementary to the homework assignment. So what I didn't want to do was create a video that, I sat here and read verbatim, homework 1 is an entirety. A lot of homework 1 is definitional in nature, and you can look at the actual text and, understand what is being talked about. What is this definition for this organelle, and what are these two concepts, and how do they differ from one another? So what I wanted to do for the video component of this is make it more complementary to the assignment. And in order to do that, I thought it would be best to focus on two areas that students seem to struggle with the most. First is the genetics puzzles, and those are largely Punnett squares on this assignment. And then 2, focus on meiosis, 1 and 2, and a little introduction to nondisjunction. So we'll start out with the genetics problems, largely Punnett squares, and then we'll move on to meiosis. So let me share my screen here. Do that. All right. So this is the first genetics problem in the homework assignment that we come across, question number 8. And you can see here, this is what is given for the problem. And what I want to talk about is my strategy in tackling these. So the first thing you're going to want to do anytime you come across any sort of genetics puzzle is, what information is given to me in the question? And largely that will be the parent genotypes. And from those, you're going to be able to answer the actual questions in the problem. So in this problem, we have pea plants, round seed R is dominant over wrinkled. Round seed is represented by capital R, wrinkled by lowercase r. And then it says pure breeding, round and wrinkled plants are crossed. Please answer the following questions. So what are going to be the genotype and the phenotype of the first generation of plants that result when we cross a round seed plant with a wrinkled plant? And so first, we have to understand what pure breeding means. And I took it to mean that the parent plants are homozygous genotypes, meaning, the round seed plant has to have a genotype of capital R, and the wrinkled plant has to have a genotype of lowercase r. So in order to solve the problem, now that we actually have the genotypes of the parents, we just need to run a simple Punnett square, and that's down here. So here's the genotype of one parent, here's the genotype of another parent, and then all we do is say, okay, capital R, lowercase r, this is going to be the genotype of an offspring, and we do that for all 4 of these squares here, and we can see that it's going to be capital R, lowercase r. Now the question is, what is the genotype of F1 plants? Well, if it's capital R and lowercase r, that is the genotype. And then that means their phenotype is a round seed. Why is it a round seed? Because the question states that round seed is dominant over wrinkled. So any genotype which has a capital R in it, i.e. round seed, means that that organism is going to be phenotypically expressed as a round seed. And so looking at these 4 squares here, we see they're all the same genotype. And so therefore, all of the offspring, i.e. F1 plants, are going to have this genotype, and they're all going to be round seed. So that's sort of a very basic introductory question, utilizing Punnett squares, very straightforward. And most students doing the problem set seemed to understand this and get this. But I just kind of wanted to start here so we understand the basic functions of Punnett squares and how we want to go about problem solving them. And, I won't get necessarily, into the exact specifics of, okay, what do we do now? Moving further through the question, this is something that students can take a look at the text. And, I think I've written it out in such a way that it's, understandable. So at your discretion, you can take a look and sort of see how we problem solve these ones. Moving on to the next type of, or I should say the next question that, utilize Punnett squares as a way to solve the problem. Very similar to the previous one. The problem is giving you what the parental genotypes are going to be. And from there, you're going to be able to determine what are the offspring genotypes. And then once you've determined that, if there's further questions about, what percentage is an offspring likely to have, such and such genotype, you can easily just look at that Punnett square and do that calculation. 1 out of 4, 2 out of 4, 3 out of 4, all of them, and that was sort of what was asked, further in these questions as well. All very straightforward once you actually have the Punnett square laid out here. And so sort of what I wanted to focus on is the questions utilizing the Punnett squares start to become a little bit more complex because there is the potential that one of the parental genotypes can be one of two things. And so we need to figure out which of those things that parental genotype is. And I'm namely talking about when a trait is, dominant over another one. So this is a good example here. Polydactyl is a dominant trait caused by gene P. Polydactyl mother has offspring with a non-polydactyl father. Their 3 children have the following phenotypes. Child 1 is polydactyl, child 2 is not polydactyl, and child 3 is polydactyl. So this is the information given to us in the question, and then here are what they want us to answer. And so here I've gone ahead and answered it. Mother's genotype is, capital P, lowercase p, and then the father is 2 lowercase p's, and this is what the child's is. Now, this is where it gets a little tricky, right? So the question states that the mother is polydactyl, and in order to be polydactyl, there needs to be at least one capital P, i.e. allele that makes up her genotype. Now, it can be capital P, lowercase p, or it can be capital P, capital P. The question is, which one is it? And this is where it's a little bit tricky. So you can see here, I've generated 2 Punnett squares. And this one is if we assume the mother's genotype is capital P, and this one's capital P and lowercase p. And we'll know which one is correct based on what possible offspring genotypes result from the parental genotypes. And so in this question, we know that one of the children is not polydactyl and 2 of the others are. And so that means that when we run the Punnett square, we need to ensure that one of the possible genotypes of the offspring allows for a child to be non-polydactyl. So looking at this one to start with, this is where we assume the mother's genotype is both capital P. We can see that any possible outcome of this results in the child being polydactyl. There's no possibility here that an offspring is non-polydactyl, but the question shows that in fact one of the children is not polydactyl. So we look at our next Punnett square. This is one where the mother's genotype is heterozygous, right? Meaning that they have the dominant allele and then they have the recessive allele. But because, polydactyl is dominant, even if mom has one copy of the gene that isn't polydactyl, she has one that does, and therefore she will phenotypically be polydactyl. Father, we know, is non-polydactyl. It's recessive, so he has to have two copies of the recessive gene in order to not be polydactyl. So running this Punnett square, we can now see that there's a 50% chance that the child will be polydactyl, 50% chance that the child will not be. So we therefore know that the mother's genotype has to be capital P, lowercase p, because it allows for the possibility that one of her offspring is going to be non-polydactyl. So that's a little bit step up in complexity in terms of doing the problem is, you know, which one is, you know, what is the parental genotype? And the only way, or I should say the best way to tackle it, in my opinion, and the way that I tackle these problems, is to just run trial Punnett squares, right? So just practice, just practice doing the, you know, potential possibilities, and then see what results from them, and is that align with what is given in the question. All right, so then we want to move on here, and you know, now we're getting into another step in complexity. So here we're talking about two different kinds of genes. So we're talking about both, you know, the color coat of the organism's fur as well as their vision. Even though we're talking about two genes now, it's still the same process. We're still going to run a Punnett square. We're still going to tackle the problem the same, which is what is given in the question. And I like to write it out. I like to, as you can see here, it's like this has got to be the genotype of parent one, and this has to be the genotype of parent two. This is simply determined by the information given in the question. Once we know these two things, we can run the Punnett squares. Once we have the Punnett square, we can answer the question. So this one, what is the chance of this cross yielding an offspring with the genotype of, you know, B, lowercase bb and lowercase nn, i.e. homozygous for both recessive copies of the allele? And the answer is 0%. And we can see that because of the Punnett square, there's no possibility that this is going to happen. And then we look at this one, and it's the same question, just now with a different genotype. And we can see, okay, there's going to be a 50% chance that it's going to end up as this here and here. This is slightly different. Got to pay close attention. This is where, you know, the color coat of the, you know, offspring is homozygous, and this is where it's heterozygous. And the question's asking for heterozygous in terms of the color coat, and we can see that's only 50%. So again, the big strategies that you're going to want to utilize when doing this is what's given in the question, write it out, run trial Punnett squares, and ensure that everything, you know, fits together logically. I want to focus on this question here. This was question 15, and I don't think that anybody who attempted this question got it correct in its entirety. People understood the logic of what they were supposed to be doing, but they overlooked a couple things here, namely what's in question A. And so I'll read the question here. The term epistasis is used to refer to a situation in which the expression of a gene is influenced by another independently inherited gene. In labs, the gene B, capital B, determines how much pigment is made. The dominant allele B results in black fur, and the recessive allele B results in brown, i.e. chocolate fur. Separate gene E codes for a protein that determines whether the pigment is deposited in the hair or not. Pigment deposited is dominant, so capital E, and then lowercase e is recessive, and that means the pigment is not deposited. So question A, what genotypes are possible for a black lab? And there are 4 possible genotypes possible. And this is one of the questions that students didn't get it correct in its entirety. Most people answered just 2 possible genotypes, but there are in fact 4. And so as I wrote, there are 4 possible genotypes for a black lab. Since B is dominant, there needs to be at least one copy of that allele. And by definition, a black lab means that the pigment was deposited. So there needs to be at least one copy of the dominant allele E. And so because we're talking about dominant, again, it's like the genotype could be both capital B, capital E, or it could be capital B, lowercase b, and capital E, lowercase e. And so you have, given that, 4 possible combinations, and I've written them out here. And that's pretty self-explanatory. Most students just guessed 2, and it was, 2 out of these 4, and they didn't identify that in reality, because we're talking about 2 genes, both of which are dominant, that there are ultimately 4 possible or 4 possible answers. All right. So moving along here, what genotypes are possible for a chocolate lab? There are 2 possible genotype combinations for a chocolate lab. This one's a bit different because in order for the phenotypic expression of chocolate fur to be expressed in the organism, it's recessive in nature. So you need 2 copies of the chocolate fur gene, so 2 lowercase b's. And because of that constraint, that means that there are only 2 possible genotype options. Again, because we know the fur is chocolate, it by definition means the pigment was deposited. So you need at least 1 copy of the dominant gene E, but you can also have 2, and both will result in the pigment being deposited. But again, because it's chocolate fur, it is recessive, and so they need 2 copies of that lowercase b, and we can see this is the answer here. All right, then I want to move on to question C. So this one was actually a little bit tricky, and I think that's in part because of how the question was worded. I don't think we were given all the information, so we need to make a little bit of an assumption here of what yellow fur means. And as I wrote, I'm going to assume yellow is the result of the pigment not being deposited. And so that's where it gets a little bit tricky, right? So as I state, with that in mind, the genotype, this question is a bit ambiguous and lacks clarity around the nature of the yellow fur. We're going to assume that that means it's not deposited. With that in mind, the genotype for the black fur lab is homozygous for both sets of alleles, capital B, capital B, capital E, capital E. We know this because the question stipulates that information. The genotype for the yellow lab is less clear. The reason being that since the pigment isn't deposited, we don't know what their color fur genotype was. It could have been, a chocolate or a black lab, but we don't really know. It's not stipulated in the question because the question just states that they have yellow fur. They don't say what it would have been had the pigment been deposited. And so, because we know the pigment wasn't deposited, the genotype for that is lowercase e, and I've written that here, but it's stated they're homozygous, so it's either capital B, or lowercase b. If they had said it had been heterozygous, then the 2 possibilities would have been capital B, or capital B, lowercase b, but we just don't know. We don't know what would have been because the pigment simply wasn't deposited. And so we have to run 2 different Punnett squares based off of what those possibilities might have been, whether it was chocolate fur or black fur. And in doing so, we'll get the answers to the questions. But again, because it was ambiguous, with the yellow fur lab, we just don't know. And so the way I approach the problem is that really there are 2 possible answers here. One assuming it would have been black fur, and one assuming it would have been chocolate fur. And again, all of this information is going to be included in the text component of homework 1. And students can take some time and look at that and digest it. I think I've written it out in a way that's easily digestible and understandable, but I did just want to talk a little bit about the strategy in terms of attacking these problems. Once again, you just always need to identify what is given in the problem, write it out, and then run trial Punnett squares. if there's any sort of ambiguity in terms of what the parental genotype might be, and based on what possible offspring genotypes there are, you'll get your answer to the questions over, what the correct parental genotype is, or what the percentage chance of, offspring having a certain genotype, etc. So that's pretty much it for the genetics problems. They're relatively straightforward, but I just did want to touch base on them. Now I kind of want to shift gears, and I want to move on to another topic that students, I think, historically struggle with, which is the process of meiosis. And I put together a little presentation here, so let me get that going. Here we go. Let's flop. All right, so we're going to focus on meiosis 1 and 2, because really meiosis is, a two-step divisional process. And then we'll talk a little bit about non-disjunction and how that relates to the process of meiosis. All right. So what even is meiosis, right? It's a related process to mitosis, but it is different. So it's a special type of cell division in, sexually reproducing organisms that ultimately results in the formation of what we call gametes, i.e. in humans, sperm or egg cells. And when we talk about meiosis, we're really talking about a phase process, and within each phase there's multiple subphases that we'll get into, but really it's broken down into two big things: meiosis 1 and meiosis 2. And ultimately what's going to happen at the end of this entire process, i.e. once meiosis 2 has concluded, in diploid creatures like humans, the end result is going to be the creation of 4 haploid daughter cells that each contain, again in humans, 23 chromosomes. So when we say diploid, what we're talking about in the case of humans, for example, is human beings have 2 sets of 23 chromosomes, right? One set of 23 chromosomes come from the maternal parent, and another set of 23 chromosomes come from the paternal parent. And so in a non sex cell in a human, the number of chromosomes in that cell is 46, and that's can be described as diploid. And what we call as a non-sex cell is a somatic cell. And in a sex cell, i.e. sperm or egg, the number of chromosomes is half of the total, and that's 23. All right, so that's essentially a broad description of what meiosis is. It's this separate but related process where we're going to create the necessary sex cells that allow for sexual reproduction in organisms that utilize that. And I'll primarily be referring to human cells because this is human genetics. All right, so this is meiosis 1, and there's several subphases to it. There's interphase, which is not technically a part of meiosis, but I wanted to talk briefly about that, and we'll get into it. It precedes the process of meiosis. It's necessary for it, but isn't technically considered a part of it. Once interphase concludes, and again, we'll get into that, we move on to the subphase of meiosis 1, which is called prophase 1, then metaphase 1, anaphase 1, and then telophase and cytokinesis 1. And we'll go through each of these in a little bit more detail here. So first, we're talking about interphase. It's kind of like a prologue. It's not considered a part of the process of meiosis or mitosis, but it's a prelude to them. It's necessary. Excuse me. So interphase in meiotic division, not mitotic division, not mitosis, but meiosis, all right, because that's what we're focusing on here. Interphase can be broken down into 2 distinct subphases, G1 and S phase, i.e. synthesis phase. There is a third subphase here known as G2, but that is limited to mitosis. G2 in meiosis is most, similarly related to prophase 1. But I did just kind of want to make that distinction there, that there is a little bit of a difference in the interphase between meiosis and mitosis. So I'm just going to talk about these things at a relatively surface level. This is really review from, cellular biology. And this is a human genetics class, so we're not going to get into great detail over the biochemistry and what exactly is taking place. We're just going to sort of talk big picture things here. What is making up each of these subphases that make up meiosis 1 and 2. And so again, interphase in G1, right? The cell is continuing to function normally, but it's, going to start preparing for this process. And the cell begins to grow in size. It increases the number of proteins and organelles, all in preparation for moving forward with the process of meiosis. If the cell is signaled to continue, it's going to enter what's called the S phase, the synthesis phase. And this is necessary for both meiotic division and mitotic division, mitosis and meiosis. So the cell in S phase is essentially replicating its DNA and doubling it. So in a cell that's going to go through the process of meiosis, normal human cell, even after it completes the S phase, and this can be a little bit confusing for people to understand because, we're saying broadly speaking, S phase, the DNA it's replicating, it's doubling, but the number of chromosomes don't actually change at this point. So a standard human cell has 46 chromosomes in it. And even when it's preparing for meiosis and it completes the S phase, it still only has 46 chromosomes. But now there are 92 chromatids. That the chromatids, right, is the replication that has happened. But there's still just that one centromere. So we haven't doubled the number of chromosomes, but we have doubled the number of chromatids. A little bit confusing. And in homework 1, I talk about in detail about what the difference between a homolog is and what the difference between a chromatid is. And we'll get into that a little bit here as we move further on. So let's continue. Oops. All right. So now we're entering what's called prophase 1. We've actually entered the process of meiosis 1. It's the longest phase. And the homologous chromosomes, i.e. homologs, pair up through a process called synapsis and exchange genetic information through recombination and crossing over. And I've included these helpful little images that I think do a good job of sort of showing what's happening in this phase, visually at a physical level within the cell. Right, so here's our pair of homologous chromosomes. We've got one from the maternal, one from the paternal. They've lined up side by side. You can see in each of these, it's got its sister chromatid, right? So there's still just the one centromere. So it's still just one chromosome here, one chromosome here. But each one of these chromosomes has a chromatid which is genetically identical, right, to its sister. So this is genetically identical to itself, and this is genetically identical to itself. But still, we're just looking at 2 chromosomes. We're not looking at 4. And like I said, going through this here, I'm not going to focus on great detail of what's actually happening at a biochemical level and, how these things are taking place and the spindle is formed and all of that. I'm just sort of going, through a very surface level of this. But this is the phase in prophase 1 of meiosis 1, where the processes of crossing over and recombination take place. This is where the exchanging of genetic information happens that is essentially essential to the genetic diversity of, in this case, humans, right? We have the paternal and maternal DNA, and they're teaming up, and there is the possibility that bits and pieces of it will be exchanged. And this is going to result in genetic randomness and genetic diversity, which is essential for human life. So this is prophase 1. And you can kind of see this is sort of a good visual representation of that recombination and crossing over taking place. Once we've completed prophase 1, and there are many subphases within prophase 1, but we're not going to get into them. But once this is completed, we move into what's called metaphase 1. So metaphase 1, in this cell, the spindle apparatus is forming at both poles of the cell. And again, we're not going to get into the biochemistry of that. But imagine there's these apparatuses that are going to be essential for the process that's taking place. Now, the pairs of homologous chromosomes that we have are going to move together and line up along the metaphase plate, which is sort of this equator, if you want to envision it like that, of the cell. And all of these paired up chromosomes that have now gone through recombination and crossing over, they're all going to line up along the metaphase plate in a random fashion, in fact. And as I state here, this is described by Mendel's law of independent assortment. It's all happening random. The reason for this, and I did kind of want to touch base on this, why it's random, why it's this independent assortment, is because the spindle fibers can only connect to the kinetochore on the sister chromosome that's outward facing from the metaphase plate. The kinetochore you can kind of envision as a dense complex of proteins that the spindle fibers at either end of the cell are going to attach to. And when these chromosomes line up randomly, it's all determined just by chance where the spindle fiber is going to attach to what sister chromatid, and it's all just total random. It's just totally random. It's just based off of chance and where that kinetochore is located. And again, this is going to help increase the genetic diversity of the species, right? And we're going to understand why that's the case as we move through the process of meiosis. So this is essentially what's happening at a broad level of metaphase 1. Once that's complete, we move on to anaphase 1. So now the attachment of the spindle fibers to the kinetochore on all the homologs is complete. And the homologs are now pulled apart by the spindle apparatuses and pulled towards opposite ends of the cell. Right? Humans, diploid creatures, have 46 chromosomes, i.e., 2 sets of 23, one from the maternal, one from the paternal. Meiosis, because the ultimate end goal of meiosis, the ultimate end result, is the production of these gametes, the sperm cell or the egg cell. By definition, all right, those are only going to have 23 chromosomes in them, because ultimately then when reproduction takes place, the sperm fertilizes the egg, and we've got one set of 23 chromosomes and one set of 23 chromosomes being joined together, during the creation of a new human life. So this process is describing, right, how we get to, just one set of chromosomes in the cell, i.e. haploid. We can describe it as haploid. So in anaphase 1, those homologs are being pulled apart, and they move towards opposite ends of the cell, 23 chromosomes in a human going to either end. But it's important to note here that at this point, the sister chromatids on each of the homologs is still attached. Right. So when we talked about the S phase and interphase of meiosis, there was this replication of DNA, and each chromosome, each homolog, pair of homologs, was replicated in an exact fashion. And that replicated chromosome we can refer to as a sister chromatid. And this is a key difference, I think, that a lot of students sort of overlook in meiosis relative to mitosis. So sort of at this point in mitosis, the sister chromatids are being pulled apart, but that's not what's happening here yet. That will eventually happen as we move through the process of meiosis. But at this point, the sister chromatids are still attached, right? They're still together. All we're doing is pulling apart the homologs. So, we've got a set of 23, we've got another set of 23, and crossing over, recombinations already happened, and we're pulling apart those two sets of chromosomes, but chromatids are still attached. So that's what's happening in anaphase 1. And you can kind of see this visual description, right? We still have got two sister chromatids here, but they will become separated. We just haven't gotten to that point yet. All right, this is sort of the end stage of meiosis 1, which is just part 1 of the overarching process known as meiosis. So the sets of 23 in humans, chromosomes, have moved to opposite ends of the cell, and they can be viewed as two distinct clusters. Broadly speaking, nuclear division happens, the cytoplasm divides, and the end result is two daughter cells, each with a set of 23 chromosomes. But again, there's still two sister chromatids on each of those chromosomes. All right. But now we've got two cells, 23 each, but we still have sister chromatids in them. So the process of meiosis is not complete yet. Once cytokinesis is completed and we have our two daughter cells, we move into meiosis 2. So this is this sort of second subphase of this overarching process known as meiosis. Meiosis 2. Because we've got two cells, the process is going to happen simultaneously in both of them. And meiosis 2 is more similar to mitosis than meiosis 1 is. And we'll get into that. It should be noted that there is no interphase at this point, right? We don't need an interphase. It's done already. We've already replicated our DNA. So it serves no purpose here. So meiosis 2 commences, and we move right into prophase 2, and then metaphase 2, anaphase 2, and telophase and cytokinesis 2. Prophase 2. So in both of our daughter cells now, the spindle fibers that were present during meiosis 1 form once again. And it's the sort of same mechanism, same machinery from meiosis 1 that happens in meiosis 2. So the spindle fibers reform, except now the spindle fibers on one end of the cell and the spindle fibers on the other end of the cell are going to attach to one of the sister chromatids at the kinetochore, and they're going to attach to the other sister chromatid at the kinetochore. So now we've got the spindle fibers attached to each of the sister chromatids on each of our 23 chromosomes. Once that's completed, we move into metaphase 2, and it's similar to metaphase 1, right? So now each of the 23 chromosomes with the spindle fibers attached to the chromatids via the kinetochore. Once that's happened, everybody moves once again to this sort of equator, the metaphase plate, and it's happening simultaneously in both cells. So everybody's going to line up, and we're going to get ready to split. Metaphase 2 moves on, and we enter anaphase 2. Again, similar to anaphase 1, except this time, the centromere is split with the sister chromatids being pulled apart towards opposite poles. So in anaphase 1, when the homologs were being pulled apart, the centromeres were still held together by this protein complex that, again, we're not going to get into the biochemistry of it, but they were still held together because there were still two sister chromatids on each chromosome. Now, because we moved along in the process, the centromere is actually going to split, and it's going to allow for the sister chromatids to be pulled apart. And this is happening in both of the daughter cells. And you can kind of see here in this image that process taking place. Now, this is an important thing to note. So when we talk about sister chromatids versus homologous chromosomes, homologs, one of the questions in homework 1 is, what is the difference between these two? Generally speaking, the major difference is that homologous chromosomes are not genetically identical to one another, whereas sister chromatids are. Right? Homologs is one chromosome from mom, one chromosome from dad. Chromatids are genetically identical replications of those chromosomes. However, in the process of meiosis, there's an exception to this. So at first, in interphase, before this whole thing took place, the DNA was replicated. And at that point, the sister chromatids on all the chromosomes were genetically identical to one another. But then we entered what was called prophase 1. And if you remember, in prophase 1, there was the process of recombination and crossing over amongst the sister chromatids. So the sister chromatids now, through the process of meiosis, are not actually genetically identical to one another. And that's sort of what separates this from mitosis as well, because in mitosis, which is just, we're replicating the cell full stop and, to create a new one, in mitosis, the sister chromatids are genetically identical to one another because that's the whole point of that process. This is a bit different. So at one point, even in meiosis, there were genetically identical sister chromatids, but early on in that process, that's no longer the case. So that's an important note to remember. Broadly speaking, homologs are not genetically identical to one another, but sister chromatids are, except when we're talking about the sister chromatids that have gone through meiosis already. So just an important thing to note. Once anaphase 2 completes, we move into telophase and cytokinesis 2. So we've almost completed this process here. The cell division begins again in both of the daughter cells. And the end result of that is you're now going to have 4 daughter cells, i.e. gametes, a sperm or egg cell. Each one of those 4 daughter cells produced through this process is now genetically unique to one another. Because again, there was this crossing over and recombination that happened amongst the sister chromatids. And then finally, during anaphase 2, those sister chromatids that were genetically unique to one another were pulled apart. Nuclear division happens, the cytoplasm is split. All of these things happening at a biochemical level happen, with the end result being 4 daughter cells, all of them haploid. So all of them only have 23 chromosomes in them. But each of those are genetically unique to one another. So that's sort of at a broad level, the process of meiosis. And I kind of put together this little slide here. I think that's really helpful. That's sort of just a bullet point list of what is the broad differences between mitosis and meiosis, because I think this is where students kind of get hung up, right? Meiosis is slightly confusing because it's, really there's meiosis 1 and 2. So it's really this 2-phase process. It's kind of the same. We're replicating DNA, we're forming daughter cells, but it is different from mitosis. And I think mitosis, because it lacks this 2 subphase process, is a bit easier for students to understand. I think they tend to learn that first, and then they come to the process of meiosis and it's like, hey, wait a minute, I'm confused here. So this is just sort of a little bullet point list of what's happening, right? Mitosis occurs in somatic cells, so not sex cells. There's no pairing of the homologous chromosomes. There's no crossing over. The cell is split once to produce 2 daughter cells, and the daughter cells are identical. Starts with 46 chromosomes, ends with 46 chromosomes. So it remains diploid through the entire process. Meiosis occurs in the germ cells. Homologous chromosomes do pair up. crossing over does take place. The cells split twice, once in meiosis 1, again in meiosis 2. Ultimately, 4 daughter cells are produced. They're genetically unique to one another, and all of those daughter cells can be described as haploid, i.e. having half the total number of chromosomes that a somatic cell in humans has. So I think this is a good sort of, overarching view of what the differences are between these two processes. The next thing I want to sort of get into, just at a very introductory level, it wasn't really focused on during homework 1, but it does come up during homework 2 and homework 3, and it relates to the process of meiosis, is called nondisjunction. All right, and this is sort of why meiosis and understanding the process of meiosis is very important to a human genetics class, because nondisjunction is essentially referring to the process in which there are an abnormal number of chromosomes in any of the given daughter cells. And when there are these abnormal number of chromosomes, it leads to major problems, So nondisjunction in meiosis refers to either a failure of homologs to separate during anaphase 1 or the sister chromatids to separate during anaphase 2. So nondisjunction can happen at both stages. And the result of these nondisjunction errors result in what we call aneuploidy syndromes. That refers to having that abnormal number of cells in the chromosome. So a common, some of the common disorders that human infants can actually survive to term with that result from these nondisjunction mechanisms are things like trisomy 21, which is better known as Down syndrome, trisomy 18, and several others. There's only one known monosomy disorder that's survivable in humans, and that's Turner syndrome, i.e. X monosomy. And I'm not going to get into necessarily what the specifics of these diseases are, or why, monosomy, X monosomy is the only survivable one versus why some of the trisomy disorders are survivable. These things were covered in further assignments, which I do believe other students are doing their projects on. So I'll leave it to them to get into those specifics. But the whole point being that nondisjunction is the result of an error of separation in either meiosis 1 or meiosis 2. And ultimately, that can result in the produced cell to have an extra chromosome that it shouldn't have in there, i.e. trisomy disorders, or it should have, one less chromosome than it should have. And those are the monosomy disorders. And you can see from here in my slide that having an extra chromosome is much more likely survivable by a human infant than having, one less chromosome. And something I found interesting in doing my research on this is aneuploidy is the most common cause of miscarriages, right? I found that interesting because I myself have actually just recently had my first child, and I have a lot of friends who are, having kids or trying to have kids. And I've known people at a personal level who have gone through a miscarriage, as I'm sure many of you have as well. And you hear about miscarriage and it's this terrible, sad event. But I never personally understood exactly what the mechanism was for that happening, why the infant, didn't survive or couldn't survive, and i.e. resulted in a miscarriage. And as it turns out, it's due to these errors, to these nondisjunction errors that happened somewhere along the way in, the meiosis process. Depending on, the error, sometimes it's more likely the maternal, germ cells were the most likely culprit. Other ones, it's more likely that, came from the paternal side of things. And we can, I could go on and on about, why as a human female ages, there's a greater chance that the, infant may, that it may end in a miscarriage, or that the child may be born with these genetic, disorders like Down syndrome. And it all has to do with these non-disjunction errors and the likelihood that they will happen as either parent ages, etc. But like I said, I'm not going to get super into the specifics here. I'll leave this to the other students and their projects to talk more in depth about these disorders and these type of syndromes. But I really just wanted to relate them to the process of meiosis, which I think students historically struggle with, because again, mitosis and meiosis, they're similar, but they're different. One's happening, twice, the other is happening just once. And if you don't understand the process of meiosis, you're certainly not going to understand non-disjunction at, a biochemical level. So that concludes my presentation for my project. I hope you found it informative. Again, a lot of the specific definitional things that have come up in this are referenced in homework 1, that students can refer to, and I think is quite easily digestible. But these were the 2 areas that I think students struggled with the most, and I hope everybody found it helpful. So once again, my name is Eric Thomas, and this was my project. And let's stop sharing my screen. And thanks so much, everybody.